So, hello everyone. My name is Peng. I'm a postdoc at the Rockefeller University. My PI is Jonathan Casanova. So it's very glad to be here. The topic I'm going to present you is a method called BP Hunter. It aims to detect for the genome-wide detection of the human variant that disrupt the intronic branch point where splicing starts. So this work was just published in PNAS, and you're welcome to read the full text. So what we do in the lab, we are trying to discover the genetic defects underlying the severe infectious diseases. So we are collecting the patients suffering the rare infectious disease infections across the globe. And we have two branches, one in Rockefeller University, one in Syria, um, in Paris. So both of the lab, we have this hybrid mode. We have about 50% in the wet lab and about 10% in the dry lab. So sometimes the wet lab people can bring us some questions, and we can bring them some computational findings for validation. So it's really a great hybrid. One example is since the COVID-19, we collected um, severe um, severe patients globally under this human genetic effort. We are studying what is the genetic defects from these patients of life-threatening infections. So today, I'm presenting you one project from the dry lab here. So we are focusing on the branch point. If you look at this schematic, you will see the DNA from DNA to RNA to mature mRNA and to protein. But when we have this NGS data, it's from DNA. But when we have the annotation, it's mostly based on the protein, missense, stop gain, frame shift, phenomena. All of these are based on the big assumption that is nothing wrong in this process. So in this project, we are focusing on the splicing event. And the splicing happens from the branch point. And we have two different types of spliceosome, major and minor, and they are having different structure. And in both cases, the adenine is most cases, the branch point is adenine. And it's located about -20 to -40 nucleotide upstream from the 3' splice site. And its consensus has been long established. So this is the basics about branch point. And what is the fact about branch point, splicing, and the disease genetics? So when we have intronic variant, most of the time they are discarded in the step 1 of the variant filtration. So what a pity. And the spliceosome operates mostly within the intron to define the exon and the intron boundary, therefore the coding sequence. And the branch point as in the beginning of the splicing event, so its disruption will likely to disrupt the splicing and have the deleterious gene product. And so far, we have seen about 10% of the disease-causing mutations are splicing related, but I think this is highly underestimated. And through a massive literature review, since 1992, there are only about 48 mutations reported to disrupt branch point and then cause disease. And from the timeline here, you can see neither the NGS data, NGS technology, nor the BP datasets has significantly increased the discovery of the branch point variant as disease-causing mutations. So, but when we have such a BP variant, it may have very severe impact on the gene product, including exon skipping, intronic retention, and alternative splice sites. Can be de novo, can be cryptic. So what's the situation, limitation, and opportunities posed here? BP has been identified, studied, and predicted individually. So I would say they lack the integration, and they are mostly focused on a limited window. So I say they lack the coverage. And most of the time, people ignore the major and minor introns, so it's lacking the specificity. And no study so far has collated and analyzed all the available BP, so we lack the knowledge base, a very comprehensive knowledge base, and no efficient tool here, so we lack method. And in this project, we are trying to cover all these limitations, and this is a framework framework of this project, we create a database and we generate knowledge base, and we apply this tool for validation and discoveries. So for the database, there are a few data sets has been published from the identification of BP from the RNA-seq data. And then we revisit our data previously published in Cell because we have seen some patients with this DBL1 deficiency. And DBL1 is the only known enzyme that is degrading these lariats. So we revisit the data and we create a script to analyze the RNA-seq data to try to identify what are the other branch points we can find. Because if we have RNA-seq read cross the junction, we can precisely locate the branch point positions. So from this program, we identify a few more than 8,000 unique BP positions. Putting them together, we're getting more than 200,000 BP positions. And then we realized that probably we need to do something on this raw data because there are two reasons. Because this reverse transcriptase in the RNA-seq, they may introduce some micromutations, and they may have some mistakes during the transesterification between the 5' spike site and BP. So we propose this consensus-guided positional adjustment to scan for a window of 5 ±2 nucleotide of each raw data. And then the outcome we get is we got a reduced number of BP with a higher agreement with the consensus. Also, there have been some computational study to predict the BP. They are using 1 or 2 experimental identified BP, and they define a window for the screening, but they use the hard distance cutoff to define, okay, this region will skip the prediction, or they create an algorithm to skip the prediction because they are considering this part is PBT interacting with another trans element. So our idea here is probably there's something good candidate missed. Therefore, we create a very simple machine learning method. But the key here we are just looking for those highly likely BP candidates which might be missed by other predictions. So putting them together and we also put this positional adjustment, we're getting more than 400,000 BP positions. And putting them together, we are having a final data set about 546,000 branch points, as you can see at the very end on your right-hand side. And this is the motive, how they look like. After getting the BP data, we also getting the intron data, and we classify all the introns into major and minor introns because we are going to study them individually. Once we get the data, let's look at we're crossing the BP positions with the introns, and then we are characterizing them. So putting them together, we are getting a subset of BP positions aligned to all the introns we collected. If you look at the distance distribution, yes, they are mostly distributed from -15 to -40 upstream from the 3' spike site. And looking at this region, we have seen a lot of introns, they have more than 1 BP. Only about 33% of the introns, they have only 1 BP mapped. Maybe this presents some tolerance of the mutations. One is gone, I have a backup plan. But if we look at the distance from the non-first BP to the first BP, it seems that very close together. And the plan B is very close to the plan A, within like 20 nucleotide upstream. And you can see the distribution, the nucleotide composition of this BP between major and minor intron. They look alike, but then you will see that this distance distribution is very different. In the minor intron, the green line, you will see like 40% of them located very close to the 3' spike site, which is not in the major intron. And we also characterize the interaction, the energy, binding energy between the BP and this U2 or U12 as an RNA, you will see in the lower the energy, the higher affinity. So you will see in minor intron, the interaction, the energy is higher. It may represent the mechanism that in the minor intron is very different from the major intron. And when we look at the conservation score, GERP and the PHILOP, we see that a higher proportion of the BP positions are likely under the evolutionary conservation. They are having a higher proportion of positive scores. And then we also do a positional comparison between the BP motif and the essential splice sites and the random intron and exon positions. On your left-hand side, we'll see within the human population, the proportion of the variance in BP and BP minus 2 positions are much lower than the intronic random positions and almost comparable to the exons and the flanking regions in the essential splice site. So on your right-hand side is the conservation score for all the positions. Interesting finding here is because we see this BP and BP minus 2 positions are really interesting. So we put them together, we see that when we measure at least one position from BP or BP minus 2, we are getting like 86% of the BP regions are evolutionary conserved. This may hint us a collaborative role between BP and BP minus 2 to ensure a certain level of conservation over the evolutionary time. And when you look at the intronic, is mostly gathered around 0. So when we have seen this kind of characterizations, we move forward to see what kind of disease genetics has been discovered regarding on BP. So through the HGMD database and PubMed searching and the experimental evidence searching, we identified 48 of them in 43 genes underlying 33 diseases. Most of them are single nucleotide variant and also some micro deletions. 16 are homozygous, 23 are heterozygous, and 6 hemizygous. When we count the occurrence of each consequences, complete skipping seems to be the major outcome from a BP variant. So we created our software BP Hunter as a web server and also as a Python program for small-scale search for those physician scientists or for large-scale analysis. When we apply the BP Hunter to those published 48 pathogenic BP variant, 40 of them can be detected. And if you look at the gray ones, they are undetected. Along the year, you will find most of them are published in the early years before 2004. If you look at the distance distribution, you will find they are mostly very close to the 3' splice site. So let's look at this 40 first. If we look at this 40, we want to find what is the pattern from this 40. So this may give us some idea to prioritize in the future. We have some candidate. On your top row, the positions, most of them are disrupting the BP, and some of them are disrupting the BP -1 and -2. And the deletions, some of them, 3 of them delete all of the BP motifs. If we look at so the suggestion is the BP and BP -2 positions are really preferred. The next, you will see when we look at the rank of the BP, majority of them from the intron with only 1 BP. So this gives us another idea to prioritize the candidates in the future. And if there are multiple BP in that intron, the first one is preferred. If we look at the BP type, because we have experimentally identified BP, computational identified BP, and mutually between E and C BP, we see the type should not be biased at all. If we look at the supported resources for those BP, it seems we need to prioritize those variants supported by more than 1 resource. And the consensus, it seems the highest conserved motif should be preferred while a little relaxed motives should be considered as well. And if we look at their human variants, the private should be paid a lot of attention, but some of rare should be kept as well. But common ones, let's try to put them aside first. And then the conservation scores, we see a larger proportion of those having both a positive score for PhilOpe and the GRP, but we still observe some of them having the negative score in both cases. So the high conservation score should be preferred but not restricted. And we also applied the Splice AI and MM-Splice, which are two recent software trying to predict the variant having the mis-splicing effect. We find that the Splice AI, they recommending 0.8 as the cutoff. None of them is beyond this cutoff. And the MM-Splice, only one BP variant is barely on the edge. So it seems that splicing scores are so far somewhat underpowered to identify those BP variant. Also, after these 40 pathogenic BP mutations, we have 8 of them missed. And using our in-house software, Sigtailor, we align these variants by using the wild type and mutant sequence up and down. We will see that clearly they are not disrupting the BP, but they're actually creating an AG in the region between BP and the 3' splice site. So we think that this AG created in this region, they promote the binding with another transacting element that is recognizing the 3' splice site. And they shorten the original space in the between that could be bonded by another transacting element. Therefore, those variants lead to the disruption of the structural organization and the formation of the spliceosome at the 3' end of the intron. Therefore, the constitutional splicing was disrupted. So after looking back those published pathogenic BP variants, we move forward to some discoveries. As I mentioned to you in the very beginning, our lab are collecting the COVID patients suffering the critical infections. And we focus on the 30 genes of related to the type 1 interferon production. And we find one of the BP variant in stack 2, and you can see the conservation score is about 0. So we think conservation score should not be really a high factor in prioritizing such cases. And we do the experiment validation. We see a lot of misspliced product. This is from germline mutation. And we also do something on the somatic mutation. We collaborate with the lab. Sure, yeah. So this is another intronic deletion. Removed all the 3 BP potential sites. So it leads to the intronic retention. So finally, we also have this negative BP reporting system because we want to treasure these negative results which can never be published. So I want to go through the summary. And finally, thank you for your attention. Yeah, I welcome any questions.